Hello, good morning. Myself, I am BSR, botany lecturer. Today, our topic nucleotides. What is the meaning of nucleotide? Nucleotide means nitrogen base plus sugar plus phosphate. When the nitrogen base, sugar, phosphate three are present, then they are called nucleotides. If there is no phosphate, then that is called as nucleoside. Nucleotide or nucleosides are micromolecules, whereas nucleic acids like DNA and RNA macromolecules. Now nucleotides consist of sugar, nitrogen base and phosphate. Here especially the sugar is a pentose sugar. These pentose sugars are two types. They are ribose sugar and deoxyribose sugar. Ribose sugar formula C5H10O5. Deoxyribose sugar formula C5H10O4. In the ribose sugar structure, when you observe, this is the structure. So here at the first carbon, second, third and fourth, 5 carbon ring. So 5 carbons are present, but the ring is made up of with four carbons with one oxygen. So especially in the second carbon, here OH and H is present. In the third carbon also H and OH is present. Two functional groups are present. So that is why ribose sugar is not much stable. Whereas in the deoxyribose sugar, you observe is the same structure, same everything is same. First carbon, second and third and fourth, and this one is the fifth one. Here only H and H is present, whereas here H and OH is present. Here only one functional group. So that is why deoxyribose sugar, due to the presence of deoxyribose sugar in the DNA is more stable. DNA is more stable because deoxyribose sugar is present. RNA is not more stable because two functional groups are present. So this is the pentose sugar. Then we go for nitrogen bases. Nitrogen bases are two types, purines and pyrimidines. Purines and pyrimidines, two types of nitrogen bases. We just look at here purines. Adenine guanines are purines. Cytosine, thymine and uracil are pyrimidines. Pyrimidines are three types, cytosine, thymine and uracil. Whereas purines are two types, adenines and guanine. You just look at here adenine and the purines and pyrimidines comparison. In the purines, two CN rings are present, whereas in the pyrimidine, only one CN ring is present. So here dicyclic rings are present, here monocyclic rings are present. You just observe here in the purines, first position, second, third, fourth, fifth, sixth, seventh, eighth, ninth. Look at here in the first and third, seventh, ninth, nitrogen atoms are present. So 1, 3, 7, 9 position, nitrogen atoms are present. Same like here also 1, 2, 3, 4, 5, 6, 7, 8 and 9. So here also 1, 3, 7, 9. Whereas look at here 1, 3, 7, 9, here at first place and third place only nitrogen atoms are present. And first carbon and third carbon. 1 and 3. Here also 1 and 3. That is the main difference between purines and pyrimidines. When you go to adenine, you look at here adenine. Adenine, two CN rings are present, especially at the sixth carbon, amino group is present. Hence it is called 6-aminopurine. Guanine is called 6-aminopurine. Guanine is also called 6-aminopurine. So adenine is also called 6-aminopurine. Whereas you go for guanine, and the guanine at the second carbon amino group is present. In the sixth carbon, oxygen is present. So you can write guanine as 2-amino, 6-oxypurine. This is only 6-aminopurine. This is 2-amino, 6-oxypurine, guanine. Then you go for pyrimidines. Pyrimidines, look at here. In the pyrimidines, is the first and the second and the third, fourth, fifth, and sixth. Look at here. 2-oxy. Here you can write clearly. 2-oxy, 6-amino pyrimidine. So you can write easily here 2-oxy, 6-amino pyrimidine. 2-oxy, 6-amino pyrimidine is cytosine. Whereas when you go to thymine, S12, 2-oxy clear, but methylated. 2-oxy methylated pyrimidine is thymine. If there is no methyl, then that is called uracil. 2-oxy methylated pyrimidine is thymine. Non-methylated pyrimidine is uracil. That is the difference. So adenine, guanine, cytosine, thymine, uracil, these three are pyrimidines, these two are purines. And phosphate. Actually, look at here, the nitrogen base, here nitrogen base is attached to the ribose sugar. Nitrogen base is attached to the ribose sugar by releasing of one water molecule. The bond is formed. Nitrogen base with ribose sugar at the first position. Water molecule release. That bond is called clearly glycosidic bond. That is bond is called glycosidic bond. So the bond between nitrogen base and ribose sugar, that is called glycosidic bond. Because of glycosidic bond, one water molecule is releasing. Like that, the phosphate, phosphoric acid, attached to the fifth carbon, there also water molecule is releasing. That bond is here peptide bond. Ester bond. Ester bond. This bond is called ester bond. So ester bond is formed between phosphate and the fifth carbon of the ribose sugar. That is called ester bond. This is called glycosidic bond. So nitrogen base, sugar plus phosphate N-type, this is called nucleotide. So you should know what is the difference between nucleoside and nucleotide. So what are the differences between nucleoside and nucleotide? Nucleoside, clearly no phosphate. Nucleoside and nucleotide. Nucleoside, nucleotide, nucleotide. So you can clearly tell how a nucleoside means only sugar plus nitrogen base. Suppose if nitrogen base is adenine, guanine, cytosine, thymine, uracil. Here also, here sugar plus phosphate plus nitrogen base. If nitrogen base is adenine, guanine, cytosine, thymine, uracil. So sugar plus nitrogen base, hence it is called phosphate plus sugar plus adenine. So only sugar plus adenine. So hence it is called adenosine. This is called adenosine. Sugar plus guanine is called guanosine. This is called guanosine. Sugar plus cytosine, this is called cytidine. This is called cytidine. Sugar plus thymine, that is called thymidine. Thymidine. Sugar plus uracil is called uridine. Whereas sugar plus phosphate plus adenine, that is called adenylic acid. Adenylic acid. It means it is in acidic nature. Acidic because phosphate is present. Slightly acidic. It is slightly base. So guanosine, here it is called guanosine. It is called guanolic acid. Guanolic acid. Guanolic acid. Cytidine, this is cytidine, that is called cytosilic acid. Cytosilic acid. This is called thymidine, that is called thymidilic acid. Whereas it is uridine, that is called uridylic acid. So it is a nucleoside and these are nucleotides. We will discuss about later on nucleic acids, DNA and RNA. Thank you.